Hello everyone, Assalamu alaikum. It's our pleasure today to have with us Professor Sohel Rahman. On behalf of IEEE Jordan section and IEEE AI local group, I'd like to thank Professor Sohel Rahman for accepting our invitation. And I would like also to thank the audience for, attending this important seminar or webinar. The title of this webinar is AI and Advances in the Medical Sector, an Incomplete Tale. I had the pleasure back in November to attend a similar, very interesting webinar by Professor Sohel Rahman. I found that talk very interesting and stimulating, and I'd like again to give this opportunity to a wider audience to attend this talk. Again, this talk is organized by IEEE Jordan section. This is the website for IEEE, and this is the QR code if you'd like to know more about IEEE Jordan section, if you are not a member already in the Jordan section. As you know, IEEE is, a worldwide organization, scientific organization. It was established back in 1884 under the name of American Institute of Electrical Engineers. Then we have another organization called Institute of Radio Engineers. These two organizations combined and merged under the name of IEEE, starting from 1963. So the name IEEE started from 1963, but the roots go back to 1884. Jordan is part of region 8. Region 8, as you see in orange, it comprises Europe, Middle East, and Africa. Professor Sohel Rahman is joining us from region 10. Region 10 is Asia, that's shown to the red of this map. So we are part of region 8, and we are happy to have our speakers from other IEEE regions. This diagram shows the membership growth of IEEE members in Jordan, and last year we achieved a record of 3,587 members in Jordan section, and this record is more than 900 than the previous record achieved the previous year. In Jordan, we have 6 professional chapters covering wide spectrum of technical aspects, and we have also 3 affinity groups: we, women engineering, life members, and young professionals. Regarding student branches, we have 23 student branches, and the last year alone, we opened 4 student branches. These are the technical chapters in Jordan, and we are happy to have wide spectrum of interest from our students. This session is organized by the local group. It's called IEEE Jordan AI, Machine Learning, Deep Learning, Computer Vision, and LLM group. This session is led by myself, Musa Al-Akhras. I'm also the chair of Jordan section, and my colleague, Dr. Hisham Al-Muaqqat. And this is the QR code for the group if you'd like to join the group later on. Now let's focus about our talk and our speaker. The title of this talk is AI and Advances in the Medical Sector, an Incomplete Tale. Now we are happy to have with us the distinguished speaker, Professor Sohel Rahman. He's a professor of the CSE department of the Bangladesh University of Engineering and Technology, BUET. He had worked as visiting research fellow of King's College London, UK, during 2008 till 2011, and again as a visiting senior researcher fellow there during 2014-2015. He is a senior member of IEEE, a member of American Mathematics Society and London Mathematics Society. He's also a peer review college member of EBSRC UK. He has been honored as distinguished member of the ACM for stabilizing achievements across computing fields. He has also been recognized as the distinguished contributor of IEEE Computer Society for his technical contribution to the profession and the society. He is a fellow of the Bangladesh Academy of Science and Bangladesh Computer Society. He received several scholarships and fellowships, including Commonwealth Scholarship, Commonwealth Fellowship, ACU Titular Fellowship, University College London Big Data Institute Visiting Grant, London Mathematics Society Visiting Grant, etc. He is also a recipient of the Bangladesh Academy of Science Gold Medal and UGC Award. He has led research and development projects funded by British Council, UGC World Bank, ITC Division, Government of Bangladesh, and BOET. He has so far published 120 peer-reviewed international journal papers. Among his notable results are the work on multi-ResNet res unit and improvement over the UNet, which is the de facto architecture for medical imaging segmentation task. High dimensional knapsack problems, sequence alignment problems, data structures and string combination. Sufficient conditions for humanicity, machine learning based prediction in bioinformatics, and meta-heuristic solutions for hard problems. Okay. Professor Sohel Rahman is also an editor of a neural network journal, academic editor of PLOS ONE, associate editor of BMC Bioinformatics and BMC Research Notes, and had edited special issues as guest editors in Theoretical Computer Science, Journal of Graph Algorithms and Applications, Journal of Discrete Algorithms, Fundamental Informatica, etc. He has also served as a program committee member in a number of conference series of international reputable journals. He regularly writes reviews at Mathematics Review and ACM Computing Review. He currently holds the appointment of a distinguished speaker of both ACM and IEEE Computer Society. He has been included in the Stanford Elsevier list, highlighting the top 2% of the most cited scientists across the globe. So we're very happy to have you with us, Professor Sohel Rahman. This is the talk, short abstract about the talk, AI. Maybe I leave at this point, maybe I leave it to you to talk more about this talk and introduce it to the audience. Thank you again, Professor Sohel Rahman, and we're very happy to have you with us today. I'll just give you the co-hosting so that you can share your screen.
Thank you so much. I'm going to share my screen.
Yeah, just a second. I want just to give you the co-host. First of all, I will stop the sharing from my side. Yeah, I stopped the sharing. And now you are the hosts. Please.
Okay. Thank you so much for your very generous introduction. Can you see my screen?
Yes.
Yes. Thank you. Let me just make the control floating so that, okay, fine. So thank you very much again, Professor Musa, for your very generous introduction. As has already been told, the title of my presentation will be AI and Advances in Medical Sector. And as you can guess, within a short span of time, you can only describe severely incomplete detail. So what I'm going to do is I'm going to talk about some of the things that we have done from my research group, just 2 or 3 things, and we will discuss in general some other things as well. So the weapons that we use from the artificial intelligence domain for the medical sector, which is of course non-exhaustive, are here. I will talk about 2 different categories of very interesting things, which we have applied in the medical sector. The first one is the CRISPR-Cas9 technology, and the second one is the COVID-19 scenario. So during the COVID-19 scenario, we did some interesting works, which I believe you would find also interesting. And CRISPR-Cas9 is a very, very hot technology, genome editing technology. In both cases, we have used AI and machine learning. For the CRISPR-Cas9, we also worked on both sides of the story, that is called on-target and off-target prediction. I will talk about those things a little bit later, which will be clear by then. So I will start with the CRISPR-Cas9 technology. Just to give you a very brief primer, I'm not going to go into the details of the biology, but the main idea is that when let me try to take a pointer. Okay. So when a virus attacks a bacteria, what happens is it sort of injects its own DNA within the bacterium DNA. But there are a lot of process of this injection and so on. But I'm going to talk about the main interesting fact, that is, when a bacteria is infected by a virus, what it does is it makes sort of a recording within its own DNA. So next time when the virus attacks again, the bacteria now knows the DNA of the virus, so it can now cut out the DNA so that it cannot infect it properly, okay? So this wonderful biological technology, biological tool has been repurposed by the scientist so that we can use it for various gene editing concepts, various gene editing use cases. So what we do is we design a sgRNA, single guided RNA, which we will insert within our system. And then there is a Cas9 complex. With the help of this sgRNA and Cas9 complex, we cut the area of the DNA that we need to remove. And then sometimes we keep the DNA disrupted, or sometimes we insert a very healthy DNA within itself. I will talk about it later again. So this gives us a very, very important and useful tool. So we break the double-stranded DNA, and then we either keep the DNA disrupted by removing only the problematic part. Sometimes, if we are lucky, we can insert a donor DNA, and we can have a completely healthy DNA within ourselves. So this is how the system works in a nutshell. So what we are going to do is we need to carefully design this sgRNA so that it goes to the right place. And with the help of this Cas9 complex, it can cut the right place of the DNA. That is the main goal. So what are the usefulness? As you can guess, we can have engineered plants so that there are many bad effects of plants, like we can remove various problematic genes from the plant so that it is not that when pest comes, it is not affected that much, and so on. For healthcare, we can have many genetic diseases. I will talk about one particular diseases towards the end of my slide. And we can have precision medicine and so on. And futuristically, we can have like designer babies and so on. And this is not at all futuristic anymore, because there is, in fact, a case where a Chinese scientist, in fact, applied this technology on human embryo. And at the end, he tried to change the gene of that gene that is susceptible to AIDS. And he said that he was successful, and a twin was born. But the problem is he didn't take ethical approval and so on. So this created a huge fiasco in the scientific world. So there are available clinical methods. So we can have genome-wide CRISPR screening. We have PCR-based methods. And there are challenges in these clinical methods. First of all, this is very costly. There are low sensitivity and limited scalability. And this is where we come in. So the prospect of machine learning and deep learning comes here. And what we can do is we can have a predictor which is fast, inexpensive, and scalable, and which can act as a primary filter for the clinical method. So the goal is that we will identify some guided RNA as a candidate for further checking through the clinical methods. So that is the main use case of AI and machine learning in this context. So this is what we have done so far. We developed a number of sgRNA predictors. First one we published in 2017 in PLOS ONE. In 2018, we did some advancement over the earlier one, but it was unpublished. Then finally, we again published a predictor in BMC Bioinformatics. I'm just going to very quickly go through this and then go into the second part of this talk, where we will talk about the off-target effect. So these are the three pipelines that we have used. We have only used sequence-based features. And here, for this particular pipeline, we only use traditional machine learning. And this was very interesting to us, because through this pipeline, what we were able to defeat the deep learning pipelines which were available in the literature at the time. So this is like usually what happens during a traditional pipeline. You need to do some feature engineering. We did that feature engineering. Then we do the data set splitting. Then we train a random forest. In one pipeline, we use the extremely randomized trees. And we did scaling of data and so on. And finally, we did train a support vector machine and so on. So at the end, we got very, very good result. We had a publicly available data set, which was a small data set at the time. So it had only 17 genes. So only around 5,000 sgRNAs, and only 20% of them was true instances. So it was imbalanced data, but the results was quite good. So this was our latest result, which was published in 2020. So we got a 91% accuracy. And because this is an imbalanced data set, we had to look forward to very good score for F1. And as you can see, we did much better than the available predictor. So this is what we call the on-target effect. And for this on-target effect, we also had a very large data set where we had 17,000 level sgRNAs on 4 different cell types. And in this, for this particular data set, we had some deep learning predictor available in the literature, which is called deep CRISPR. But we are able to able to defeat the deep learning pipeline, except for only one data set, which is called HEC 293. And later we found out that this data set has some problem in it. So overall, we were very excited that we have been able to defeat a deep learning pipeline with a traditional pipeline for this very, very hot topic. But the second stage is that we need to consider the off-target effect. So far we have talked about predicting an sgRNA, right? So we want to predict an RNA sequence, which will at the end, which will at the end give us and take us to the right place of the DNA so that we can cut the DNA and replace it with, replace the problematic DNA with a healthy DNA. So part of the healthy DNA. But the problem is, which is called the off-target effect. So what happens is when we send the sgRNA within the system, it sometimes go to some regions where they don't match always. So here, for example, in this case, this sgRNA has matched with a DNA sequence where there are 4 mismatches. So normal, the rule is that A will match with T, T will match with A, G will match with C, and so on. But here, as you can see, A has matched with C, here A has matched with A, here C has matched with C, where it should have been G, and G has matched with A. So there are 4 mismatches, and what happens is these are non-specific, and this could have unwanted effect and adverse effect. So this could alter the genome and so on. So this is called the off-target effect. So the scientist wants to know whether a given sgRNA candidate has this sort of off-target effect possibility. If it has very high possibility of an off-target effect, then we should not use this sgRNA as a candidate RNA for the genome editing case. So this is now a lot of research effort are being spent on this particular case. So for example, we had score-based methods, which are not very good. We had traditional machine learning methods as well in the literature, and then came the deep learning methods. And among the deep learning methods in 2018, they had this CNN-based method. Then there was the deep CRISPR I mentioned. Deep CRISPR was able to work on both settings. So it can work on target setting as well as off-target setting. We had CNN, CRISPR, and so on. And as you can see, they have this sort of components within their system, like they had embedding. Sometimes they have used pre-training. Sometimes they have used CNN, feed-forward network, LSTM. Sometimes they have applied attenuation as well. And in some cases, they have some non-specific features as well. So this is the state of the art of current literature. So the data set, if you talk about, we have used a data set which was used by someone in 2018, a very comprehensive data set which used 2 different cell lines. There were total 30 sgRNAs and a total of 153,000 samples. But as you can see, only 656 positive samples. So this is a hugely imbalanced data set. So the imbalance ratio is 1:230. Anyway, we had to work on it. So we split the data set into standard split, like training set for 60% training set, validation 20%, and test set 20%. So this was our proposed pipeline. We focused on a lightweight and interpretable pipeline. That was our main goal, because interpretation is very, very important in this context. So we will use the raw sequences. The sequence encoding will be done. I will talk about very briefly about the sequence encoding. Then we will use the generic RNN-based model, and then we will tune the hyperparameters, identify the best model, and then we will try to predict. This is the sequence encoding that we have used. This is basically one hot encoding imposed one over the other, and there is one extra line for direction. Here, direction is based on the precedence of the ATCG, these 4 nucleotide characters. And overall, the idea is to superimpose the one hot encoding of sgRNA and the candidate sgRNA and the DNA pair from the data set where the sgRNA matches the DNA. So this is how we do the encoding. And this is our generic model. We supply the input, that is the encoding, within the recurrent layer. We could have optional recurrent layers, 2, 3, and so on. Then we have 1 or more hidden layers. And finally, we will give the output. We have used vanilla RNN, we have used GRU, we have used LSTM, and so on. We have used different options like stacked RNN, bidirectional RNN, and so on. And we had to go through a very rigorous hyperparameter tuning, because we had various hyperparameters, and without tuning the hyperparameters, we cannot do well. So for hyperparameter tuning, it came out as a very, very difficult exercise for us, because we work in a constrained computation setting. We don't have enough GPU to run a very exhaustive hyperparameter tuning exercise. So what we did, we applied a genetic algorithm. So we had an initial set of model parameters. Then for each model, each mode, we set a hyperparameters were selected randomly from possible set of values. So we use 20 models in our genetic algorithm. Then this is the main bottleneck step because we have to train the model, which takes around 30 minutes sometimes on NVIDIA RTX 3090 GPU. This is the computational model that we had only. We didn't have any higher computation power. We use the cross entropy as the loss function, and we use Adam optimizer. So after training, we check the fitness of the model. In our case, for this, our genetic algorithm setup, we use the AUPRC as the fitness. Why AUPRC? Because as I have mentioned, it is a highly imbalanced data set. So we had to use the precision recall curve. And based on this fitness, we rank the models, and then we select the models for the next generation. And here we use the genetic operators like crossover and mutation. We use a tournament selection with tournament size 2. Crossover and mutation was performed on selected model by pairs. And we had an elitist version of the genetic algorithm, which worked very well, where only top models were kept for the next generation. So as I have already mentioned, we have used crossover and mutation. So for crossover is done on pair of models at a time. Each hyperparameter is selected with a random probability of 0.3 for crossover, and hyperparameter values are swapped between the pair of the models. Each crossover step returns 2 new models. That is the next generation of Spring. And for each new model, where we also applied mutation on it. So each hyperparameter is selected with a random probability for mutation. So new value for the selected hyperparameter is chosen randomly from the possible values. So after mutation, we have 20 set of model parameters for the next generation, and the process continues with model training. We could only use 20 iterations because, as I have already mentioned, the model training is a bottleneck step. It takes more than 30 minutes sometimes, and so on. So this is the result of our hyperparameter tuning. If we had used all possible hyperparameter combinations, that would give us 72,000 plus combinations, which would have taken 1,000 days in exhaustive search with the setup we had. But with the help of genetic algorithm, we very intelligently searched only 0.6% of the total space. And we did independent search on 2 types of encodings, 3 types of RNNs, and 2 genetic algorithm variants, plain and elitist. So the best RNN, LSTM, and GRU models were finally selected. So this is the hyperparameter values that we got for the different RNN setup. So finally, some results. Here we can see that we have compared with almost all state-of-the-art tools, all deep learning tools. Just in one case for deep CRISPR, we could not recode it because they didn't provide all the retrained and recoded it. We use it as an off-the-shelf model. All the other models were retrained and tested within our setting. So we found that our LSTM and GRU performed very well, and it outperformed all the previous studies. And it was very interesting to see that the genetic algorithm very intelligently searched only 6.6% of the possible search space, which in the end helped us to get the best model in our setting. And we improved our AUPRC by around 6% and AUROC by around 11%. This is not the end of the story because, as you know, whenever you got a better model, you can quantitatively get good results, but it's very important to see whether the results actually supports actually can explain the model. So we focused on the interpretation of the model. We have used a very state-of-the-art technique called integrated gradient machine. So we found very interesting things. For example, mismatch at the PAM region is not tolerated. I did not talk about it explicitly yet. So this is the sgRNA, and the last 3 nucleotides are called PAM. So this is always fixed. This is why this is never any change in this region is never tolerated. And we found out that mismatches are dominated by TG and CG. And we interestingly found 2 subregions within the seed region. And we found that this is somewhat supported from the literature as well. So these 2 points are supported by the literature. So we were confident that our machine was able to match the existing knowledge base, existing explanation of the literature. And this is something we found new. And this is why we have divided the seed region into two regions, seed 1 and seed 2. We found that after some studies that in 2017, Zhang and others suggested a sensitive sub-region of seed, which they named as core region, which basically overlaps with seed 1 region. So the new finding that we have found had some interesting evidence in the literature as well. So all the details can be found in this paper, which has been published in Briefings in Bioinformatics. And if you have any questions, I will try to answer the questions after the presentation. So now, just to give you motivation, why should we work on CRISPR-Cas9 technology? So to the right, I have an infographics, which is very important. And this is, I mentioned that I will talk about one particular disease, which is called sickle cell disease, which is a hereditary disease. So suppose Susan has the sickle cell disease caused by a single DNA letter mutation, which is here. So what we can do now is in the lab, we can create a strand of guided RNA. The guided RNA that I have shown you the models to predict this guided RNA, okay? So we can create a guided RNA that matches the mutated DNA sequence. So this guided RNA, with the help of Cas9, which is called the molecular Caesar, these two complex can be sent through an injection within Susan's body. And then what happens is these guided RNA and this molecular Caesar, that is the Cas9 system, will identify the problematic area of the DNA. It will go there, and the molecular Caesar, that is Cas9, will cut out the affected sequence, and then the scientist can insert a healthy DNA sequence to replace the clipped sequence. So what happens is Susan, with her problematic DNA, through this process, can come to a scenario when she doesn't have that mutation anymore. And when she doesn't have that mutation anymore, she doesn't have sickle cell disease anymore. So this is very, very interesting. And for sickle cell anemia, already I think the clinical trial has been completed. So I just wanted to show you this. This is like a very simple string algorithm, string problem, like we have twinkle, twinkle, big star. But we know that big is a problematic word here. So we can cut out big and put little instead of big. So we get twinkle, twinkle, little star. Very simple. Okay, so if you are not yet motivated, just to motivate you a bit more, in 2020, Emmanuel and Jennifer got Nobel Prize in chemistry for the discovery and development of CRISPR-Cas9 genome editing. So with this, I finish the first part of my talk. Now what I'm going to do is I'm going to just very briefly talk about what we did during the COVID-19 period, okay? So here, the motivation was to give a platform for the scientists to start working on. At that time, there were many papers were being published, and many misinformation or disinformation were being propagated and so on. So we thought that we should have a knowledge base for COVID, which can be used by general public as well as scientists when they want to do something, do a new innovation or discovery and so on. So where do I start? So this was our answer. We wanted to create a portal which will have the knowledge base for COVID-19. We call it COVID-19 base 2. So we invited the scientists to start from here. So what we had in our portal, we had different entities like disease, drug, gene, long coding RNA, miRNA, protein, and so on related to COVID-19. Okay. So in addition to that, we wanted to record the interactions and associations between these entities like disease drug association, disease gene association, and so on. But the problem is it is very, very difficult to read the infinite literature, right? And identify these sort of associations and entities and so on. So what we wanted to do is we wanted to automatically infer these interactions and associations. And we also wanted to evaluate how confident we are in this automated inference, and whether these inference, through these inference, if we find out some drug for a particular disease, are there any drug side effects? So this was the main idea. And to do that, we have used natural language processing. And we also tried to have a sort of a manual curation effort and so on. So what we did, we used a Kaggle dataset, which was called the Cord 19 dataset. It was based on 138,000 papers from PubMed, bioRxiv, and medRxiv, where they have searched. This dataset was prepared by Allen Institute for AI, and it was available at Kaggle. So they look for these sort of keywords like COVID-19, coronavirus, SARS-CoV-2, MERS-CoV, and so on. So this was our main data set. And we also use some dictionaries like for gene, we use HGNC. We all know about HGNC who work in this area. We use MIR-base, we use LNCpedia, we use PDB for the protein, we use disease ontology, we use drug bank, and for side effect, we use the CIDR database. So the goal was to extract disease drug interactions and disease gene associations. And then we will also extract drug protein associations. And finally, we wanted to extract side effects for drugs. So I'm going to just talk about the first one because we don't have enough time to cover everything. So this is what we did. We on our data set, this is what we applied. We find disease and drug names present in the literature that is on our data set. We applied a dictionary-based approach. Then what happened is we extracted the disease and drug pairs. So we collect sentences that contain the disease drug pair. It was a co-occurrence-based approach. Then we also go through an anomaly detection exercise. So because there are some mistakes, so we did a k-means clustering. And if there is an anomaly, then we remove that pair. If there is no anomaly, we go to the next step. So in the next step, what it is, we did a sentiment analysis. So the idea is whenever we find a disease drug pair, the question is, it is a positive pair or it is a negative pair. For example, it could be the inference of that paper could be that this drug is not good for that disease, or the inference of that paper could be that this drug is good for that disease. So we did a sentiment analysis. And for the sentiment analysis, we have used a off-the-shelf text blob model, and we also use this unsupervised model. And from this unsupervised model, we got sentiment trait. From the text blob model, we got a polarity. And from the disease drug pair extraction, we found out the minimum distance between the disease and drug term. These three we have used as features and train a neural network model. This was a slightly deep neural network model. And from this neural network model, we identify the level of each disease drug pair with a confidence score. So through this exercise, automatically, we were able to identify more than 2,000 drugs, more than 1,800 diseases, more than 1,900 genes, and so on. And we were able to identify more than 22,000 disease drug associations. Among those associations, there were more than 21,000 positive associations and 1,300 negative associations. Similarly, for disease gene association, we were able to identify automatically almost 16,000 associations, and for disease mRNA 56, and for drug PDB 194 association. So I'm going to just point out for COVID-19, we also identified COVID-19 specific associations. For example, for COVID-19 drug, we were able to identify at that time from the literature 514 associations, among which 492 were positive, 22 were negative. So I'm going to discuss 4 interesting use cases which we were able to automatically identify from the literature. For example, dexamethasone. So COVID-19 base identified dexamethasone as a positive drug, as a useful drug for COVID-19. And the machine was 77% confident. And it was also found effective against pneumonia, respiratory failure, diarrhea, and so on. And from other studies, we were able to find out that it reduces the risk of death from COVID-19 from 40% to 28% for patients on ventilators, and from 5% to 20% for patients needing oxygen. So our machine, COVID-19 base, were able to positively identify and effectively identify dexamethasone as a good drug against COVID-19. Similarly, our machine also was able to identify ivermectin as a positive drug for COVID-19 at the time, and we were around 78% confident. And it was also found effective against pneumonia and diarrhea. And we found that in other studies later, it was also found very useful for COVID-19. Similar case was found for remdesivir, and in this case, we were around 70% confident. Very interestingly, at the time when we were working on this project, hydroxychloroquine, you might remember that it was thought to be an effective treatment for COVID-19 for a certain period of time. But our knowledge base, our automated machine was giving us negative results. So we, with around 65% confidence, it was saying that hydroxychloroquine will not be effective for COVID-19. And later, it was found that it is really true that hydroxychloroquine had some other side effects. And this is why it was not later used for COVID-19. So to sum up, we had some errors as well, which I'm not going to present here, but the error, we can understand that it is due to the inherent limitations of the methods and approaches adopted. For example, we have used a off-the-shelf machine, off-the-shelf sentiment analysis, and so on. And this is why we provided confidence level. So when someone is working with something, and if it founds from our system that something is 70% confident, he should take it with a grain of salt. Or maybe if it is 50% confident, then he should take it with a grain of salt. And we also, what we did is we also provided the link to the source manuscript with the GOI so that he can check himself whether our system has made some error and so on. We also had a feedback mechanism so that any scientist who would find some problem with our inference, he can give us feedback and so on. We wanted to incorporate drug trial information. We were not able to do that at the time. We did continue to do some periodic updates, and we published another paper later with the latest updated knowledge base, which was COVID-19 base 3.0. And we always thought of manual curation, but without funding, this is very difficult. So all the details can be found in this paper. So I will wrap up very quickly because I think we want to go through some question answer session as well. So as you can see, in 2024, the Nobel Prize in Physics were given to computer scientists, right, for foundational discoveries and inventions that enable machine learning with artificial neural networks. Again, in chemistry, the Nobel Prize was given for computational protein design. So we can, in fact, not we, many people are discussing the actual winner in these two cases are artificial intelligence. So we are patiently waiting for artificial intelligence to win in the medical sector in near future as well. So I don't think it is very far when we will have computer scientists or people who have been working in medical sector to win the Nobel Prize very soon. So this is the end of my presentation. If we have any question, I will try to answer your question. Thank you very much for your patient hearing.
Thank you very much, Professor Sohel. And thank you for the attendees. If you can allow me to kick off and start the questions. I think this is a very interesting talk. Can you just brief us about the requirements if someone, because among us there are many, young researchers who would like to join this field. Any kind of advice or roadmap how to, enter this very interesting and at the same time not easy kind of research?
Yeah. I think I'm sure that you have you know very well that to work in machine learning, it is very important to understand how the machine works, and it is not. Now we are at a stage. There are two. I should say two new directions that we now have to cover. Like, for example, when we used to work in traditional machine learning, what we used to do is we used to sort of do some hand engineering with the features. Like, so we used to talk to the domain experts and probably get an idea about which feature should be used for this particular case, for example, disease prediction and so on. But when we have come to the deep learning era, now feature engineering is done by the machine itself, right? So this is where the domain expert comes in. And when feature engineering is done by the machine itself, it looks like a black box. And when it is a black box, many people do not want to believe in it, right? So a few years ago, for example, when we are more focused on traditional machine learning, we could have applied the traditional machine learning without knowing much of the details of the system, much of the details of the algorithm. But now it is very important to understand what the machine is doing, how it is using the feature, how it is identifying the features and how we can interpret the features. So this is why I think the first thing that our younger aspirants should do is learn the system very well. So not just applying. So this, I know this applied machine learning field is very, very popular. We want to apply machine learning in everything. And if we apply machine learning in a data set, we always find something interesting, even if it doesn't mean anything. So this is why my first suggestion will be to understand the models, understand the algorithm. How does CNN work? And how does CNN identifies the features of a picture, of an image, to identify an image, something like that, okay? And then, of course, you have to be good in programming, right? You have to do all the experiments. You need to understand how things work, and optimization as well. And the second aspect now is green computing. Now, people are questioning, for example, OpenAI, that what is the carbon footprint of training OpenAI versus, for example, DeepSeek, right? Because DeepSeek is being trained with much less time, and you know what has happened in the last couple of weeks. So this is what my suggestion for the younger kids and younger aspirant researchers would be: to learn the system very well. And to learn machine learning, you need to know the mathematics behind it, and you need to understand statistics as well. And then you can take informed decisions as well, which model should work well, and so on. And then, of course, you also need to have the capability of coding very efficiently, so that you have like a very green scenario. Okay.
Thank you very much. Do you have any other questions from the audience? Please go ahead. You can start the mic and raise your question. Dr. Iman, Jude, I can recognize a few names here. Yeah. Okay. Again, I have two more questions, but I will allow the audience if they have questions. It was very interesting to me when you said that one of the models, overperformed deep neural networks. Do you have any, explanation? If you can please elaborate on that, because with deep neural networks, we always have the issue of limited resources, and we need high computation power. But if we can have a solution that can achieve a similar result, but with less computation power, that would be, I think, that is something of interest for a wider audience.
Yeah, exactly. In fact, we had a very similar reaction from a reviewer of that paper, because it was very surprising that why the traditional machine learning model was able to outperform a deep learning model. But anyway, the thing is, probably it might be the case that the deep learning model that has been used, for example, for that project of on-target effect, the deep learning model was being used. They have used a CNN model. Maybe they haven't used the true potential of the deep learning pipeline. That would be the case. But we were able to outperform the deep learning model, and we were the point that you have mentioned, that is perfectly true, that our training was much less involved, and we were able to work in a very, very restricted computational setting. But at the end of the day, and this is why, let me tell you this, our archive paper has a connotation. Archive paper has a subtitle that beating the deep learning model, but the reviewer suggested we should not write that, because it probably we haven't been able to beat the deep learning model per se. Rather, we have been able to beat a particular setting of deep learning model.
Yeah, thank you. One last question, because we send the invitation from people from the engineering and IT background, and also from medical sector. Now, as someone coming from, IT or engineering background, regarding the biological aspects of things, do you think, it's better to have, a team of biologists in the same team, of research team or so? Any guideline or any suggestions?
No, I truly believe that we should have a medical people from medical field in the team. The problem that we have in Bangladesh is that the medical doctors, for example, they are so busy. It's very difficult to get their attention in this sort of research work. And you know, this is why the research work I have been doing, it is not like with the data set of Bangladeshi people, you know, I have used a public data set, which has been published or have been used by other researchers. So it is, I truly believe, for example, the interpretation exercise that we have done on our second work, I think it is still incomplete, because we could go further and try to identify in a clinical setting whether that sort of sub-region have any inner meaning, for example, if you may. But we couldn't do that. We couldn't take that step. If we could take that step, probably we could have submitted the paper in a more impactful journal. You see what I mean? So this is important to have someone who is a domain expert.
Yes, thank you very much. We have a question from Jude. Hello, Jude. Thank you for joining. Please go ahead.
Good afternoon, everyone. How are you? Professor Rahman, thank you for your wonderful lecture. It was very insightful. My question is, I do believe you heard about the explainable AI, which is recent advancement, helping to eliminate the black box concept about AI, maybe helping physicians or medical personnel to trust AI models more. So based on your two research, where do you think using explainable AI would fit if it could be used in these researches?
Okay, not only in the research that I have done, but overall, now, as I mentioned, explainable as AI or interpretability of a model is extremely crucial. And the good thing is that nowadays we have some interesting tools, I would say, which can be applied to the deep learning models, which otherwise were treated even 2-3 years back as a black box. For example, people are using SHAP or Shapley, you probably have heard about it, to interpret deep learning models. And for traditional machine learning models, we used to have like this sort of feature ranking or some sort of internal scoring of the traditional machine learning models, like SVM could give you a score, and you could have used recursive feature elimination technique within SVM to identify the top features. So if you are diagnosing a disease, you could have identified the features that have helped you identify that disease, and then you can match with the knowledge base of the doctor and the literature whether the identified features actually match with the knowledge base, or if there is something new, you could have talked to the doctor to see whether this makes sense as well. So this is very interesting. And in my case, for example, the two I have discussed for, I briefly mentioned about the interpretation exercise we did for the CRISPR off-target technology. As I mentioned, we identified an interesting region. We are still not sure how impactful that is to identify a region which is not tolerated. But when I say not tolerated, that basically means that this region will not have the off-target effect that much, okay? So this might be useful when you are checking the sgRNA in a clinical setting, because in a clinical setting, when you are doing it, you can probably avoid this part from your check, which could be less costly and so on. And for the NLP work, where we try to provide a knowledge base for the scientists as well as general public, here we did provide the actual source of our inference, so people could go to that particular DOI, to that particular paper, and check whether our inference was right or not. So through this, we tried to provide the interpretability in that work.
Thank you very much.
Thank you very much.
Thank you, Jude. Do you have any further questions from the audience? Please go ahead. Please seize this opportunity if you have any questions for Professor Rahman. I think we're done with the questions. Dr. Sohel Rahman, thank you very much for, accepting our invitation. Thank you for your time. I know the time zone is different in Bangladesh, so it's 3 hours, to the east, so that it's like 9 o'clock in the evening at your local time. Thank you again and for your patience and for joining us. we're happy to, to have you with us today. And on behalf of IEEE Jordan section, we thank you for your contribution. And if you allow us to post the recording to our official channel so that future researchers can look at this who were not able to join, can attend the recording.
Please do so. Thank you very much for inviting me, Professor Musa. It was indeed a pleasure for me to give a talk, and I hope that we will continue collaborating in this way.
Thank you very much. Thank you for joining. Thank you everybody for attending, and have a nice evening. Take care.
Thank you very
Bye-bye.
much.
Bye-bye.
Bye-bye.
Thank you very much.
Assalamu alaikum wa rahmatullah.
Salam.